explain what a lutefisk actually was?
Oh, if those of you who don't know, lutefisk is I think it's a Norwegian delicacy that consists of dried cod soaked in lye, with some spices, I'm sure.
How's that? Great. Hold on, do we have to switch? I'm not coming in here. Oh, I know, I think it's my fault. Oops. Hold on. That's better. Ooh, I got email. All right. Cool. So let's see. So is that Oxford? Okay. So I'm going to take you from, I guess, the more rarefied world that NAMD lives in to the more hands-on, messy world that you're going to live in for the next 2 weeks. So this is about structure building. It's about how you actually get your protein from the web, where you see a nice cartoon picture of your protein, think, I want to simulate that, to NAMD, to where you can actually simulate it, get some new results, publish some papers, hopefully, that kind of thing. Great. Okay, so one thing I think that's worth asking right at the start is, why are we doing this at all? Why do we have to spend so much time building structures? One thing people often say is, I thought PDB files had structure in them. They've got residues, they've got connect records. There's a lot of information in PDB files. Can't we just use that? Okay, that's a valid question. I think the real answer to that is that there's more than one way to simulate a biomolecule. Different programs use different ways to represent the force field. The charm force field that you saw in Jim's talk is just one way, but there are others that we're not going to get into because it's only 2 weeks. And so the process of structure building sort of encapsulates the decisions that you've made for how you want to represent your protein in simulation, mapping it from what I would consider to be really a more abstract representation of the protein in the PDB file to something that's fully concrete, including all the terms in the force field, the PSF file, that you need for NAMD, because NAMD can't just make those up on its own. Another reason for structure building, maybe you've heard this before, if we knew what we were doing, it wouldn't be research. That's kind of true for simulations, because it's very easy in simulation, actually, to get to build a structure that no one's ever simulated before. And that's often what you want. You want to be able to have complete control over what you're simulating. This probably clips. In order to, because you can't just take a PDB file, throw water around it, and necessarily have an interesting system. Usually you have to do a little bit more. Like you'll have to do a SMD on it, or you'll need to, build a new kind of lipid around it. This is what research is about, and so that's why you need, I think, this is why scripting tools and having full control over your structure building process is going to be useful to you in your work. Okay, so let me just give an overview of what the structure building process looks like so that we don't lose the forest for the trees. So this is ubiquitin. You're going to see a lot more of ubiquitin over the next few days, I think. The first step is, of course, to get a structure until we've managed to solve the protein folding problem. The actual atoms in the structure look kind of like this. It's kind of a mess, but there's something missing. It's the hydrogen atoms. So the structure building first process is just to add atoms that are missing in the structure. There might also be missing residues, missing side chains, that kind of thing. And to a certain extent, NAMD can build those in for you based on the ideal information that's in the topology file. I'll get to that later. Another thing that often happens when you're structure building is that you'll have some side chain like histidine that can exist in multiple protonation states. What do? It's up to you. You need to decide which of these nitrogens to protonate. Once you've got that, you need to decide how much water you're going to add so that you can and then maybe probably trim it down to a sphere. So I think the thing to sort of keep in mind, or the thing to think about, the way I like to think about it is that when you're structure building, we kind of work from the inside out. We sort of start with the internal components, the protein, get the individual side chains oriented right, then we start adding water and building on and so forth. Another more complex example would be the grammacidin A channel. This is just a really simple transmembrane peptide. It exists as a dimer. But in a way, this is kind of the most complex thing that you can build for something that's so small, because you've got a dimer protein, you've got a membrane, and then water around there. So you've got just about everything you could ever simulate so far, except for DNA, because I don't know DNA very well. And so what's complicated about this? Well, we get the structure, but we're only partway done. Grammacidin has these non-standard N-terminal and C-terminal residues at the end because of these functional groups that are attached. You got to build a membrane around it. That's already can be a little tricky. Membranes are cantankerous beasts sometimes. And add water around it. Okay, so you've got pretty much everything that you would ever want to do in a simulation represented in this little, I'd say, 3,000 atom structure. So things can get complicated in a hurry. All right. So general strategy. What's the big picture here? Figure out what you want to simulate, of course. Working from the inside out, we're going to build our individual components, add missing atoms, modify ionization states. If a protein has, say, a salt bridge inside of it, we're going to have to tell the structure building program that this salt bridge has to be built, and so forth. Once we've got everything, individual components built, we're going to combine them all together. VMD has very simple commands for doing this, and then get to a nice minimized energy state so that we can equilibrate. Okay. So the tool that you're going to use is a tool called PSFGen. This is a component of VMD. It's just another set of commands in the scripting language of VMD that you're going to import into VMD and then use along with the rest of VMD's features. So what does it do? Well, it kind of starts from this. The abstract representation of the protein would be just the sequence of residues. And so this is going to link your residues up to form one long chain, one peptide chain. It's going to combine, say you had a multimeric protein like the ATPase system, which contains more than one chain. It's going to combine those chains into one structure. We're going to add those salt bridges or different functional groups that I alluded to before. If you've got missing side chains, you can usually guess those. Missing hydrogens, it's very good at guessing missing hydrogens. And then write out the structure files that you can use in your simulation. Okay. Why do I keep harping on scripting so much? There's a scripting language built into VMD. It's called TCL. There's also a Python scripting interface, which some people like. This is really what allows you to build almost any structure you can imagine is with this scripting language, because you can use the ability of VMD to select arbitrary groups of atoms to move things around, to rotate things. Basically, you don't need a set of windows. You have everything. The scripting language will allow you to express any complex structure within VMD. Okay, so kind of how it looks like. Just you'll see a lot more of these scripts later in the hands-on NAMD tutorial later on. But you're going to be importing this PSFGen package into VMD using this command called package require PSFGen. And what's nice about having PSFGen built into VMD is that you can then intermingle commands that deal with only structure with commands that deal with how VMD looks at the protein. And so we can use these atom selections in VMD and combine them with commands that have to do with structure building in a very flexible and useful way. Okay. This is, I think, probably the most interesting slide, because this really gives you, I think, the whole picture of how all these files link together. We have the PDB file, and then I mentioned that you're going to, if you have a structure that contains multiple segments, you split those up into different files, each one for each connected segment. That's going to, you have your topology file, which is just basically a bunch of recipes for how to build each kind of residue. Combine those into PSFGen, you'll get a bunch of PSF files. You might run this again to get a second PSF file, a third PSF file. Combine, and you have something that's ready to run in NAMD, except that you don't know how the bonds and angles that you built from the topology file are supposed to be calculated in NAMD, and so that's what this more general parameter file is for. This will not make sense to you, I think, the first time you see it, but when you have all your files, you have your parameter file and topology file all sitting in one directory, it helps to look back on this and say, okay, how do these all relate? What's this for? Okay, and this is just what I said about what all the files are for. Okay, just to, now when you actually start building, what you're going to end up doing is, as I mentioned, splitting the structure into segments. Basically, let's take the BPTI example, or the ubiquitin example, actually. This is ubiquitin as you would get it from the web. You have protein, which is sort of the cyan and blue segments, and the little red dots around here are actually water that are contained in the crystal structure. You might want to use those in your simulation if you think that the crystallographer got the positions of the waters right. Sometimes they do. Crystallographers are good. So, but the first thing that you'll need to do is to split those into two pieces, one containing just the protein, one containing just the water, because water doesn't ever form any bonds to proteins, so that's why they're separate. Okay. Sometimes actually proteins, crystal structures contain water or contain hydrogens if it's a really low, like sub-angstrom resolution structure. And there's a few of those out there. NMR structures usually contain hydrogens because that's what the NMR guys actually see. But despite all that great work in finding the hydrogens, it's probably best just to blow them away. And that's because no one ever names the hydrogens the same way. Hydrogen atoms in the PDB that you're going to download are almost always differently named than what is in your charm topology file or whatever force field that you're going to be using. And so just to avoid problems later, it's easier just to delete them and let PSFGen build them in for you. You can also get name conflicts with things that are not hydrogens. Like, for example, if you had some protein where one atom was named C1 and the other was C2 in the PDB file, but they were switched in the topology file. And then what you would see, if you load it into VMD, you'd see this nice kink in the chain as it goes from the top to C2 to C1 and back down. This is one of the good things that VMD is good for. You'll see this right away when you build your structure, and you can say, I need to change the names so that it builds the structure in the intelligent way. Okay. Dealing with unknown residues. Does anyone know what this is? Anyone recognize this group? You might have heard of it. It's only recently found to be important. This is the 21st amino acid. This is selenocysteine. This is the selenium group up here, and the rest is just the regular amino acid part. You might want to simulate something that contains selenocysteine. Who knows? But you might find, oh no, my old charm topology file from 1997 doesn't have it. What am I going to do? Well, this is why we have the session on doing ab initio calculations, charge assignments. I think this will be on Friday, where you'll learn how to deal with completely novel, I think. Where you'll learn something about how to deal with groups that aren't necessarily found in a generic charm topology file. Sometimes you get lucky. Sometimes you can just take a piece of one residue definition and a piece of another and just stick them together. And sometimes they're even meant to work that way. For example, lipid recipes, sometimes they'll give you the lipid tail and the lipid head group, and you're just supposed to combine them. That's fine. That's the way it was parameterized. But other times, yeah, you actually need some quantum chem to get the charge assignments right. Otherwise, you'll just have nonsense. Okay, it happens more often than you think. As I mentioned, the grammacidin A peptide has not only these formyl groups at the N-terminal and C-terminal ends, but also it has these residues with alternate chiral symmetry than what normal residues have. And so basically just by analogy with existing structures, you can rename some of the atoms and build it anyway. So it can be not too bad. Okay, like I said, if atom names are switched, you'll see it in VMD right away. So just go into the file and swap them. Okay, so that was all building. That's actually usually the hardest part. Once you have that, once you have all the individual components built, it's usually just a matter of writing some standard VMD scripts to take a PSF file from one component, combine it with a PSF file from another component, get a bigger PSF file. Not too hard. The exception is water. Since water has to fit around your structure, has to fit around your system, we've provided a special package called Solvate, which surrounds your protein with water, finds overlap, builds the water structure as big as you need. It starts from a water box that's been equilibrated in an NPT ensemble, which we felt was the most applicable for what people would be using it for. And so once you build the water around it, you basically have equilibrated randomized water that's ready to go for your equilibrium simulation. Okay. Just to give you an idea of how what this looks like when you combine it with VMD's structure building commands. Another way to do this, if you don't want to use Solvate, is you can just throw a big box of water around your protein, around your membrane protein in this case. So this is just basically overlaying and combining two PSF files into one. Chop the water that you don't need. Oops. And basically, this is what Solvate is doing for you. It's going to it can build the water box around your protein. It can delete the waters that would otherwise overlap with your protein in your membrane, and then tell you how big to simulate the periodic boundary conditions in your system, so that you have a continuous periodic system containing water. Okay. That's about it. When you've got your system ready, then you're ready to go on with the things that Jim, I think, told you in very nice detail about, minimizing it, performing equilibration, and then finally being ready for some free dynamics. And I think you'll learn about analyzing the equilibrium dynamics in the next session. So that's it. Thanks. There is a question here.
Yeah. So when you do this Solvate command, the water which is used around the solute, do you need to do the equilibration solvent and solute fix?
Holding the solute fix or the solvent fix?
Solute.
The solute fix. Yeah, I would recommend it. The way that I would recommend it would be to keep the solute fixed until the water has equilibrated around your protein, because otherwise you could. Solvate's actually pretty good about not putting things too close to the protein. It shouldn't put things so close that it's going to distort your protein structure when you first turn it on. Even so, the paranoid way to go is to keep it fixed until everything else is equilibrated. Another reason to keep things fixed, or at least constrained, if you're doing a constant pressure simulation, the simulation cell might be changing rather rapidly during the initial equilibration phase, and you don't want your system to be your protein to be unduly affected by that. And so really only once everything else around the protein has stabilized, what I recommend taking constraints or fixed atoms off of the structure. Yeah, another question.
Not possible to produce mixtures of different solvents?
Solvate doesn't do that. I think that you could adapt it to do that. One other thing that Solvate does not do is add ions to your system. It's not that hard to do. You can just replace an ion with another once you know the distribution of ions around your protein. Typically, right, but I mean, so it kind of depends on how precise you want to be. Do you want to compute Poisson-Boltzmann around your protein? Do you just want to put enough ions in there to stabilize it? So it's in principle possible to do, but yeah.
Two questions. First, can we use Solvate to do simulation, but not in water, for example, in ethanol?
Sure. It would be easy to do, actually. It'd be easy just for me to help you with this, because the script itself just takes a pre-equilibrated bath of water and replicates it. And so if you had an equilibrated bath of ethanol, it'd be very easy just to adapt it to Solvate and do a Solvate ethanol.
And my second question is, can I use VMD as an interface to, in a sense, equilibrate myself to structure? Because I want to modify protein, for example, choosing one residue, and then manually equilibrate the position of this residue because I know I have some feeling of which position is going to have.
Right, okay, so you want to
modify, for example, torsional angle manually and things like that?
For modeling the side chain, I mean, it depends on how. It's a little bit awkward in VMD to model the internal backbone conformation, If you're just going to mutate one side chain to another, especially if the new side chain is going to be smaller, it's easy to, there's a command called mutate in PSFGen, which will just change it, and then it'll build the new mutated side chain instead of the original side chain. If you're going to be manipulating backbone conformation, this is actually something where I think a more sophisticated modeling package, and I can give you names, would be better at it. So if you don't actually know, VMD and Solvate and PSFGen kind of assume that you've got a pretty good idea already of what the backbone conformation of your protein is, either from a crystal structure or something else. It kind of needs to be an input into VMD. And so once you have that, then I think you could place your side chain in a reasonable way. Does that answer your question? Or? Okay, we can talk during the coffee break or something. Somebody else? Yeah. Yeah. Cavities? Yes, it will fill in basically anywhere it can find a water, it'll put a water. But so what you often have to do is go back, figure out which waters need to be removed, and one more easy 5-line script in TCL can remove those extra waters. So yeah. Anybody else? Yeah.
Does NAMD also run on microchips?
Yes, I think so. Is that right?
Yes, it does.
But it's still a little.
It might crash the new version of the operating system. But
Okay. So Mac OS X is still kind of a moving target in some ways, but I think we're getting more stable and
But yes.
yeah.
Yeah, runs on the old.
On 10.2, is there a version of Mac OS X that it's pretty good on?
The current release one runs fine as far as I know, but the
10.2.6?
latest builds have been knocking over the new build on.
Okay.
Yes and no, we're working on it.
All right.
Okay, I just wanted to tell you that when you come back in, please pick up your first tutorial, VMD molecular graphics. So there is now some